Okay. Good morning. So welcome. Today we are going to finish up with our skeletal muscle. We're going to briefly mention smooth muscle exists, but then we're going to get into our heart stuff, which is going to be the rest of this unit. Okay. So last time we were talking about our skeletal muscle fibers. So the final thing we want to piece on is a little bit about fatigue in the different types of muscle fibers. So we know we have fast versus slow twitch fibers. We know we have oxidative versus glycolytic fibers. And we know that we can combine those two things together and describe three different types of fibers. And we talked a little bit about recruitment of the different fibers and recruitment of motor units in movement. So now we're talking about fatigue, like when you get tired. Okay? So we're going to start with high intensity activity. So if you're working out really, really hard, high intensity activity, sprinting, lifting really hard or something, this is mainly using your glycolytic fibers. So fast glycolytic fibers are quicker to fatigue than other types of fibers. Okay? This is because they're relying on glycolysis, that anaerobic, not using oxygen way of producing ATP, which creates a buildup of lactate, which we often talk about as lactic acid, causing a sort of burning sensation. So that is one thing that kind of causes fatigue. Another thing that's happening at the same time, those glycolytic fibers, they don't need oxygen, so they can be greater in diameter. They're thicker, they're bigger, they're chunkier. So they don't need to get oxygen into the center of those cells, which means they also have more sarcomeres in parallel, which means they're stronger. All this to say, when we have really strong contractions, they're bunching up. They actually compress the blood vessels that run next to them. So they're actually kind of cutting off their own blood flow in some cases, which long-term is a problem if you cut off your blood flow too long. So this is part of why high intensity activity causes fatigue more rapidly as well. In really extreme cases, with high intensity activity, you can undergo something called neuromuscular fatigue. So that's actually coming from the nerve itself. It's basically that you're running out of neurotransmitter. So acetylcholine is our neurotransmitter going to skeletal muscle through our somatic nervous system. So if you deplete that acetylcholine because you've been putting out so much neurotransmitter as you're firing those neurons over and over and over again, this can cause fatigue from that sort of control side. So at that point, you feel like you can't even contract at all. There are also some alterations in enzyme activity that are related. Recovery from this takes minutes to hours after high intensity activity. So this type is fatigue. With lower intensity activity, we take longer to fatigue, right? So we know that this is relying on those oxidative fibers. We know that oxidative phosphorylation produces more energy, but it needs oxygen. We know those fibers are a little skinnier, a little smaller. They're going to contract a little more weakly. We can use them for a longer time. The reason they eventually do fatigue is usually they're running out of like glucose. Much of their glucose is coming from glycogen stores. So if it runs out of glycogen, that can start to cause fatigue. And this takes a longer time to recover from. If you get to the point that you're fatigued by the low intensity exercise, takes a while to build back, for example, that glycogen. Another cause of fatigue or lack of fatigue is really just your mindset. So this is not necessarily to do with a muscle or a nerve specifically, but they do say that a will to win, like a race or something, really does kind of control certain amounts of fatigue you can push through. Some people can, some people don't need to that much. Okay. So here we have a picture of fatigue. You may or may not have seen something like this in a physiology lab, but what we're just looking at here is our stimuli from our nerves, right? So here we're looking at action potentials. And we can see they're spaced out, kind of slow, right? So we can see we're producing tension and then we're relaxing and then we're producing tension again, okay? So this isn't really causing fatigue because it's spaced out enough. You have like little bits of rest in between these stimuli, okay? By contrast, this next one, we see those action potentials coming faster together. Okay, so we can see that we're at our peak tension for a while, but as this continues on, eventually we hit this fatigue point and we can't produce as much tension, we can't produce as much force anymore. Finally, we're looking at what happens if you hit a period of fatigue, rest for a little bit, and then try again. So after sitting around, waiting, resting, if you try again too soon before you're fully recovered, you may be able to hit that peak tension again, but you're going to fatigue quicker. So the time to fatigue here is longer in the second bout of exercise than this first one. Okay, so we just want to understand that sort of like high view stuff here. We're going to really be focusing mainly on the heart in this unit. Just want to make sure that we touch on these. Okay. Also, talking about types of exercise. So training does make a difference, right? If you practice, then you get stronger, you get faster. Different types of exercise, so different types of training, change your body in different ways. So aerobic exercise, so like running or something, right? This is going to increase your oxidative capacity. So aerobic exercise tends to increase your ability to do oxidative phosphorylation. So that's going to be things like increasing how many mitochondria your cells have. And we can convert some of our fibers into other types of fibers. Okay, so we're going to see that the two we can trade off. We have our fast glycolytic fibers versus our fast oxidative fibers. Okay, so some of these can kind of switch. So the difference between these two types of fibers, they were both fast twitch fibers, which has to do with the type of myosin they have. So the difference was in the glycolytic versus oxidative. So the glycolytic part had to do with having lots of enzymes for glycolysis, mainly some differences in diameter, some differences in blood flow. But the big thing is that tendency to run glycolysis over and over again with a lack of oxygen. The oxidative fibers are doing oxidative phosphorylation. So there are a bunch of differences that we kind of looked at, but really one of the main ones is going to be number of mitochondria, maybe some stuff about diameter. So you can't change the type of myosin you have. So fast and fast are the two that can convert to each other, but you can change things like enzymes. You can make more of those, right? You can change things like how many mitochondria does this cell have? So that's why some of those fibers can switch from fast glycolytic to fast oxidative or vice versa. Okay, so recapping, aerobic exercise, aerobic, you're using oxygen, you're breathing a bunch, you're trying to do oxidative phosphorylation, have this lots and lots of ATP for this sort of long-term exercise. So you're going to want to do oxidative phosphorylation. So you're going to increase oxidative capacity. You're going to convert some of those fast glycolytic fibers into fast oxidative fibers, which was that like intermediate type. Okay. You're going to increase your mitochondria, right? So you're going to make more of this type of organelle where we have oxidative phosphorylation happening. And you can change a little bit about fiber diameter and blood flow as well. Okay. So you're going to make those fibers skinnier because you want that oxygen to penetrate them better. Okay. And you can increase the number of capillaries going to those muscle fibers. So you're increasing blood flow. So this is all in response to training. If you do this over and over and over again, your body is going to try to adjust basically to increase the amount of oxidative phosphorylation that you're doing when you exercise. If you do really high intensity exercise, we're going to also see changes, but they're going to be the opposite ones. So instead of focusing on oxidative capacity, trying to do oxidative phosphorylation, you're going to try and design your body so that you can have a lot of those fast glycolytic fibers, really focusing in on strength, right? So you're going to decrease the oxidative capacity, and your trade-off is that you're going to essentially get increases in fiber diameter, which means the contractions of your muscles are going to be stronger. So they're going to get wider, they're going to get beefier because we got lots of sarcomeres in parallel. So they're also going to be stronger. Okay. The only problems with this means that you're gonna fatigue a little quicker because you have converted to these more fatigable fibers, and you are no longer going to be getting a bunch of oxygen to those cells because you're really focusing on glycolysis. So if you're not using oxygen, you don't need mitochondria. I mean, you need some, but not many. And general decrease in oxidative capacity. So this is why different types of exercise do different things to your body. If you don't exercise at all, or if you really don't use your muscles, say like you're bedridden or something, your muscles can atrophy, right? So they die off. And sometimes in lab, we see that some of the bodies sometimes end up with like really little skinny, rippy muscles. So a general decrease in size, if you're not using the muscles, is the result of like losing myofibrils, so losing muscle fibers themselves. And this also happens in some disease states, an example being muscular dystrophy. Okay. So next thing we're going to do, I am really just going to tell you that smooth muscle exists. So when we talk about it again in A&P 2 as relates to blood vessels, it is not a surprise to you that smooth muscle exists. So don't focus too much on memorizing this. I'll add to the outline like 1 or 2 facts I want you to know about smooth muscle. But basically, we have 3 types of muscle. We have our skeletal muscle that we've been talking about this whole time. That's the voluntary stuff. That's the stuff we look at in lab. Smooth muscle is different in shape, right? So everything we've been talking about with like sarcomeres and stuff, that structure of a muscle is pretty specific to our skeletal muscle. So our smooth muscle, and then as we move into our heart, our cardiac muscle, looks different, functions a little different. Okay, so smooth muscle cells wouldn't look long and skinny and fibers. We got these like teardrop things all connected to each other with gap junctions. And we're going to end up with like just a single nerve kind of lying over top of a group of smooth muscle cells, which then pretty much all fire together. Okay? So that is the general gist of smooth muscle. So when we talk about smooth muscle in the future as relates to our blood vessel, we're not going to like name different smooth muscles or something. We're going to say like, okay, this group of muscle, like this group of muscle layer is contracting as one. Okay. So they do still have actin and myosin, but they're different shaped. They don't have sarcomeres. They just look different and behave a little differently. I think that is pretty much what I want you to know about them for our purposes. Okay. Yeah, I think that's what I want. We do remember that they were one of our effector organs for the autonomic nervous system. So that's just a picture of what that looked like before it was just a word on a list when we were doing our nervous system stuff. Okay, so we'll come back to this when we do blood vessels in the future. I want us to move into the heart. Okay. So we're going to be talking about the heart, which is an organ. It is also a muscle. So we're going to be focusing on how it functions. So we're going to be focusing on how it is that it is able to push blood out through your body, how we're able to get oxygen into it, oxygen exchange to the tissues, get carbon dioxide back, get carbon dioxide into the blood, eventually get that to the lungs, breathe that out. So we're going to be talking about the heart part now at the end of A&P 1. And when we roll in with A&P 2, we're going to be talking about those blood vessels going to tissue, and we're going to talk about the lungs. So we're going to be on this topic of like how the blood and oxygen and gas work together for kind of a long time. So for our cardiac stuff, we're going to just start. The reason we did the cardio anatomy assignment was just so that we have a little overview of the major parts of the anatomy that we're going to talk about as we talk about like what's contracting, where is the blood going. In anatomy lab, again, in the spring semester, you'll have a really detailed look at the anatomy of the heart. So we're going to keep it like kind of a bit more limited right now. So just focus on what I tell you about what is functioning, and I can update those outlines to clarify exactly what I want you to know from this stuff. Okay. So our heart is important. We need blood to go places. We need to get oxygen out to our cells and other nutrients. We need to take our waste from those cells and get rid of it. We also need to pump around our hormones from our endocrine system. We have immune cells in the blood that need to get where they're going. We have all sorts of proteins that need to go different places. So the heart is pumping all those things out. Okay. So when we're talking about the heart, the first parts we want to start with really is our chambers and our blood vessels. And a note about the cardioanatomy assignment, I know that the auto grading at first was real weird. I went through and did some hand grading on those. So if you did it early, you should have seen your scores change once. I'll go and check in on those again. Basically, if it looks like you showed some effort on those, I'm going to give you credit, but I want to make sure that we know what's going on. So we're going to just recap that real quick. Okay. So blood coming into the heart from your body is coming through your vena cava. There's a superior and an inferior vena cava coming in to the right atrium. Okay. So both of these blood vessels are just collecting oxygen depleted blood and pulling it to the heart. Okay. So this is your right atrium, and we're going to call this the first chamber of the heart. There are 4 chambers of the heart. From here, your blood is going to pass into the right ventricle. And I think this is one of the things that was really not working on the key. So this is your right ventricle. Okay. So the right ventricle is down here on the bottom. This is the larger chamber. Now the right side of your heart, its responsibility to receive that blood from the body, but that blood doesn't have oxygen in it at the moment, right? So we don't want to send it out to the body right now, because we'd just be using that same old blood over and over again. That's not going to help us from a functional perspective for our tissue. So first we're going to need to send that blood to the lungs to go get some oxygen. So from the right ventricle, we go through something called the pulmonary trunk, right, which does have a valve in it and then splits out to our lungs on either side through these pulmonary arteries. And a thing I want you to note about the pulmonary arteries in diagrams, this is the one place you're going to see arteries that are colored blue. Okay, so our pulmonary arteries are colored blue because when we're coloring things blue and red in images, what we're referring to actually is not, is this an artery or is this a vein? What we're trying to tell you is what is the oxygen content of the blood in this vessel. So these are the only arteries you're going to see colored blue because they still have oxygen poor blood in them because they haven't been to the lungs yet. Okay. So we want to make sure that we don't get these confused with the veins just because they're blue. So they're going to the lungs. Anything that's leaving the heart is an artery, right? So that is actually the definition of an artery, is something that is leaving the heart. Things going away from the heart are arteries. Things coming back to the heart are veins. So we're going to the pulmonary arteries. We're going to go out to the lungs. In the lungs, we're going to be in contact with air. We're going to get oxygen into the blood, and then it's going to come back to the heart. Okay. So coming back to the heart, we're going to go to the left side now. So now, because we are coming back to the heart, these are veins, but they're colored red because they now have oxygen in them. Okay. So these two places, right, like the pulmonary arteries and veins are the one place that you're going to see those colors flip-flops from basically all our other arteries and veins. Okay. So from the lungs, we're coming back through the pulmonary veins into our left atrium. Okay, left atrium here. Okay. And we are then going to go into our left ventricle. Okay, this is our left ventricle, which is big and strong. So you can see this kind of strong muscle. You can see this thick muscle around this ventricle. This is because this is where we have to get all the pressure to squeeze the blood all the way out through your whole body. So we're now going to go out through the aorta, which then goes pretty much everywhere through its branches. Okay. So this is going to the rest of the body, or what we call the systemic circuit. All right. And a primer on how I tend to draw this as I'm doing my little scribbles. I like to make the heart an actual heart because, I don't know, I do. Okay, so this is what an actual heart looks like, but this is often how I'm just going to draw it. Sometimes I'll add in valves, sometimes not, depending on what we're talking about. So that's going to be the right atrium, the right ventricle, left atrium, left ventricle. So if you want to draw it that way for yourselves, for lecture purposes, we do not need to have anatomically correct drawings here. We're just focused on understanding the function. We can worry about what the heart actually looks like in lab next semester. Okay. So you just want to remember that from the right ventricle, we're going, I will use blue. So we're going to go out to the lungs from the right ventricle, and then we're going to come back to the left atrium from the lungs, and then left ventricle pumps out to the body. So I guess I could say lungs and then body. Should we connect them? Make it a loop. Okay, so something like that. So that's the general flow of blood through these chambers and major vessels of the heart. As we talk about how the heart functions, we're going to be talking about how and why these different chambers start to squeeze, start to contract, putting pressure on the blood, and then how that pressure causes the blood to try to run away to move somewhere else, right? This is how we get from one chamber to another. We increase the pressure, make that blood really uncomfortable, so it wants to go somewhere else. Okay? So we're going to talk about how we trigger those contractions through our electrical conduction system. And we're also going to be looking at the pressures themselves and the effect that has on some of these other features in the heart. Okay, so that is the main bit of our heart anatomy. So when we start to name blood vessels, we do have different names based on like how big, how small the direction of our blood vessels is. So from the heart, anything going away from the heart, like going straight away from the heart, is an artery. So when you see blood vessels that are thick white walls in lab, they're kind of squishy, they should sort of bounce back when you touch them. Those are arteries, right? So they're big comparatively. Arterioles are our next set of branches. Basically, we've gotten smaller. So actually quite small. You won't see arterioles in lab, really. We're never going to name an arteriole in lab. So if you're looking at it with your naked eye, it's an artery. So arterioles are going to be next. So they're going to branch smaller. Then we're going to have the capillaries, which are the site of exchange with the tissue. And then we're going to start our return to the heart with venules. So those are like our pair of the arterioles, right? Anytime you see like this sort of L on the end, this is telling you that they're small, right? Like L for little. So arteriole, a little artery, venule, a little vein. Okay. Leading into our bigger veins, so named things we might see in lab, back to the heart. This is a closed loop, so things are not escaping, not coming back mainly, right? So blood is just going all the way around. We're not leaving it pooling anywhere, right? Right. In terms of what is in our blood, we will talk more in the future about especially our red blood cells, our erythrocytes and hemoglobin. But we do want to remember that a lot of our blood is made up of plasma, which is one of our types of extracellular fluid that we might remember from Unit 1. And if we don't, that's now a great time to jog our memory on that. Okay. So plasma, fluid, solutes, so things dissolve in it. Our erythrocytes are our red blood cells. They're largely carrying oxygen and carbon dioxide. Then we have our white blood cells, the leukocytes, so that's part of your immune system. And then we have platelets, which are like little tiny flakes of cells, actually. They're kind of weird looking. They're going to be important for blood clotting, which we will also talk about when we talk about the blood specifically. So that's what's in these. And we talked a little bit in order while we're looking at the heart, where the chambers are going. But when your book talks about the cardiovascular system, they talk about it as two circuits, okay? So I tried to put this in your cardio anatomy assignment because people do find this confusing, but it's actually not that bad if you just think about it. Hopefully the pictures I picked for you were clear, is what I'm saying. So when we're talking about circuits, we're just talking about where is the blood going. So the pulmonary circuit is just talking about that loop where we're getting deoxygenated blood from the right side of the heart, sending it to the lungs, and then sending it back to the heart, this time to the left side. And that's all that is, okay? The systemic circuit is everything else. So the systemic circuit is all the blood coming from the left side of your heart, going out through the aorta and going everywhere in your body. Okay? So that is the very simple view of what these two things are. Okay. And the next thing, your book likes to use physics words to talk about these two circuits. And this is moving into the part of A&P where we do start using a fair number of physics words, just a warning. We will define them, but spoiler alert, it's coming. Okay. So when we talk about series and parallel, what we're talking about, right? A series of things is just like dominoes, right? So series in order. So if you have something in series, you have 1, 2, 3, like that. So that's series flow, right? So if you do things in an order and you could number them 1, 2, 3, that's series flow. If you have things in parallel, right? So think about like parallel lines, right? So if you have parallel lines, or in math, you have a parallelogram or something, you have two things running next to each other. So when we say something has parallel flow, we're saying that something's splitting and going out to two lines separately. So here, it wouldn't make sense to call one of these 1 and one of these 2, because we're not going first through this top dot and then through this bottom dot. We're going through them both at the same time. So this is series, and this is parallel. So when we say that something is going in series flow through the cardiovascular system, we're saying it went through this organ first, then it went through another organ. That's series flow. So for example, it's a series when we say that we have to go first through the heart and then through the lungs. But in many places, we're going to have stuff going in parallel. So like blood that's going to whatever, your left knee doesn't have to then go to your liver. It's just going to go like to your left knee and then back to the heart. Okay. So the blood flow to your left knee versus to your liver would be in parallel to each other, right? Because they're not flowing through one then the other. They're just going out at the same time and then separately returning back. So when we talk about that, that's what we're talking about. So flow, if we talk about the systemic circuit and the pulmonary circuit together, count as in series, right? So we have 1, 2, right? 2, that's series, okay? And that's because we have to go first to the pulmonary circuit, right? So lungs is pulmonary, okay? And then next, second, we go out to the body, okay? So we went back to the heart. Now the left ventricle is pushing us out to the body through the systemic circuit. Second, because we had to go to the lungs to pick up oxygen first. Okay. Now, when we say that things are going through these two circuits in series, that is all that means, is that we had to go to the lungs first, then we went to the body second. Versus here we have a map of the systemic circuit. So when I say body, when I say blood is going to the body, this is kind of what I'm talking about. Okay, so we can see that blood did first go to the lungs, but now we can see that it's going to the GI tract and the kidneys separately. It's going to the skeletal muscle separately from the skin. So if we were to trace blood to the skeletal muscle, right, you'd go to a muscle through an artery, arterial, capillary, And then you'd return venule, vein, etc., back to the heart. Okay. So that would be a separate little loop, a parallel loop to blood going to your skin, for example. Okay. So we have parallel flow to most of our organs so that you can separately control what's going on in your skeletal muscle versus what's going on in your skin. You can think about some examples. I don't have any off the top of my head, but you can think about like an occasion where you might want like lots of blood flow to the skeletal muscle, but you don't need a ton of blood flow to the skin. Actually, since we've talked about sympathetic, parasympathetic examples that we could add on here, right? So we know we have like absorptive and post-absorptive states, right? We have the rest and digest, and we have the stress state. So in rest and digest phase, you want to digest, you want to send a lot more blood to the GI system in that state, but you don't want to, because you're sending lots of blood to the GI system, also send tons of blood to your skeletal muscle. You'd want to send a bunch of blood to your skeletal muscle when you're actually running around using skeletal muscle. So in your kind of like fight or flight mode, okay? So that's why it's helpful that we have parallel flow to these different organs. Some organs do work together. So there are a couple of systems that are in series, right? So here we see you go to the GI tract and then the liver. So 1, then 2. So that would be an example of series flow here. But in general, think about the systemic circuit as all our different organs in parallel. This is a sort of side note. So one potential misconception about blood in the heart, that we do have blood going through the heart, going from ventricle to ventricle, but that is not actually the blood that's letting your heart muscle work. So your heart muscle separately needs blood that goes to those muscle cells of the heart, and that's different from the blood that is pumping around. So when people talk about like coronary arteries, or you say someone has a coronary when you're talking about like a heart attack or something, what you're referring to is not the blood necessarily inside the heart, but you're talking about blood outside the heart that feeds the heart muscle. Okay, so we're not going to focus on the names of those specific arteries here because, again, you will see those in lab next semester and focus on naming them then. But just do know for now, the blood inside the heart is designed, it's destined to go to the lungs and then out to your organs in the body. The blood that feeds the heart muscle itself is coming from the outside, essentially. Cool. We are also going to just skim through this position of the heart stuff. Again, this is stuff that would be tested more in lab. So just know, heart is in your chest, which is called your thorax. It's sitting on top of your diaphragm, which is a big flat muscle that helps you move the lungs. It's about the size of a fist. It's situated in a shiny, smooth little sac that doesn't have much friction, so that the heart can increase and decrease in size smoothly without rubbing against stuff, getting kind of stuck, causing pain. So if your pericardium, this bag around the heart, is not smooth and slippery, there's some type of disease state, that is something that can cause physical pain. So that's something you might see in a patient. We're talking about the heart walls. They're made of muscle. Okay. So we have our left ventricle over here, big and thick. We have our right ventricle a little thinner, because our right ventricle doesn't have to get blood that far, right? Like it only has to get it to the lungs. The lungs are right next to the heart. Don't need really strong muscle there, versus the left ventricle has to get blood all the way to everywhere in your body. Okay. So if we were to slice through any of these walls, we would say that there's an outer membrane called the epicardium. There's a middle portion. That's where you'll see the muscle. So this would be the word that I would think is the most important from this list for you guys. So myocardium refers to the cardiac muscle. So the actual muscle cells of the heart form your myocardium. And then there's also an inner layer of endothelial cells called the endothelium. And this is going to be true as we move forward to like all our hollow organs. They're all going to have like an inner lining of cells as well. They've got an outer lining, an inner lining, but the most important part is the myocardium where we have the muscle. We will focus on the valves a lot when we talk about the cardiac cycle. So we don't need to worry about memorizing too much about this today, but there are valves between our atria and our ventricles, so they're doorways for the blood, basically. And valves are important in your body, in the cardiovascular system in general, because they control the direction that blood can go. So they're like one-way doors, essentially, or they should be unless there's a problem, okay? So when we talk about the cardiac cycle, we're going to be focusing on that order of blood flow that we went through on a cardioanatomy assignment, and just real quick right now. We are also going to be thinking about the valves because they are going to control the blood doesn't backflow to where it was, which is important because the heart and the movement of blood is all dependent on pressures that you're creating in these chambers. A rule that I do want us to learn for now, which we want to remember ideally forever because we are going to want this again in A&P 2. Pressure is basically a gradient, right? So like when we talk about chemical gradients, we talked about rolling down a hill, right? From high concentration to low concentration. Pressure, conceptually, we want to think about kind of that same way. So we're going to go from areas of high pressure to areas of low pressure. If you think about like a hose that you're holding your hand over or like making a kink in, and then versus when you release that pressure, that can kind of help you remember it, right? Like if you build up pressure, eventually it needs to release, right? Okay, so high pressure wants to go towards areas of low pressure, essentially because we're trying to reach equilibrium or trying to reach a state where we just have uniform pressure everywhere. So this is going to be true as we talk about the chambers of the heart, and then we're going to be thinking about this as well as we think about our blood vessels in the future, as we think about our lungs, and so on. So high pressure to low pressure. So the normal direction of blood flow is what we went through on the assignment. Right atrium to right ventricle, out to the lungs, left atrium to left ventricle, out to the body. We will talk about these valves as we go through the cardiac cycle as well, but we can name them right now. So in the right side of the heart and the left side of the heart, we have atrioventricular valves, A for atrium, V for ventricle. They look different, right? So they're both between an atrium and a ventricle, so they're AV valves, but they have different numbers of like little leaflets, right? So these are like little leaflets. So on the right side, we have 3. So this is a tricuspid valve, so 3 cusps, versus on the left side, we have the bicuspid valve for 2. So bi for 2, cuspid for cusps. We have a separate type of valve in our arteries. So our separate type of valve are semilunar valves. So we have them in the blood vessel that goes out to the lungs. So in the pulmonary trunk, which is that first artery before it splits out to the right and left lungs. We have our pulmonary valve. And then we have in the aorta coming out of the left ventricle, we have an aortic valve. So these look a little different. They're like little bags kind of in the walls of the artery. They don't have little strings attaching to them. They're different than our AV valves. Okay. Yeah, we do have a second. So we're going to start electrical activity. That stuff in that PowerPoint is mainly like preview of structures that we're going to focus in on. So focus in on the structures as we talk about them in our like physiology context. pure anatomy you can get in anatomy lab. Okay. So when we talk about conduction in the heart, what we are talking about is like electricity passing through the heart, just like when we talked about nerves and action potentials. Actually, like that kind of is a form of electricity going down a nerve. So when we talk about the conduction system of the heart, we're going to be talking about these charges traveling through pathways in the heart, controlling the contraction of the muscle. Okay. So a thing to know about the heart, which is cool, and this also happens in some smooth muscle, but we want to think about it for heart cells right now. So the heart cells actually exhibit something called autorhythmicity. So some of these cells in the heart, not all of them, but some of them, are able to generate their own electrical pulses, like without any input from anything else. So like if you put a couple of these cells with this autorhythmicity into a petri dish, they would still beat. These cells would just create these charges on their own, right? Which is awesome, actually. So these autorhythmic cells that have their own pulses of electricity that they just do, it's just part of how the cells work, are going to be responsible for kickstarting the contraction of heart muscle cells, so the actual cardiac muscle. So our autorhythmic cells are going to form part of our conduction system. So conduction, we're sending this signal through the heart to contract it. And we're going to have two types of autorhythmic cells. We got pacemaker cells, and we have conduction fibers. Okay, so the pacemaker cells are usually going to be setting our pace, and then the conduction fibers are usually going to be conducting that impulse. But depending on specifically which area of the heart we're talking about, we're going to see that they have slightly different baseline rhythms. And the rule of thumb here is whatever cells have the fastest rhythm are going to be in charge of the whole thing. Okay. So that sentence will hopefully make a little more sense as we talk about like what happens when things go wrong. Okay. So our pacemaker cells have this autorhythmicity. They're spontaneously depolarizing membrane potentials that are generating action potentials, is the fancy way to say that. So what we're saying is that spontaneously, just like on their own, because of the way these cells exist in the world, they're depolarizing, right? So they're getting excited, their membrane potential is going up, right? That just happens from the way these cells are, from the way they're built. So this is automatically just generating action potentials spontaneously from these pacemaker cells. So these cells are mainly going to be responsible for coordinating and providing the rhythm of your heartbeat, right? So your heartbeat is really based on this spontaneous depolarization of the pacemaker cells. Okay, so that's the baseline for your heartbeat, mostly at rest. The conduction fibers, their main role in the conduction system of the heart, so in the like electrical part of the heart, they are going to be conducting those action potentials from the pacemaker cells out to the myocardium, so to the like cardiac like muscle cells, which are then going to do the contracting. So we can talk about the order of this next time. So just this general piece is where we'll leave it off. We're going to do pacemaker cells, conduction fibers. So next time we're going to be naming the specific types of pacemaker cells and their locations, as well as the specific types of conduction fibers and their locations. So that's where we'll pick up next time. But that's it for today.